Hello guys, this is a new series of videos. I'm going to talk about microbiology, foodborne microbiology, the microbiology related to food. In this series of videos, we are going to discuss the microbiomes. We have the microbiology of the bacteria mainly, and then a little bit of how the bacteria affect the food we eat in terms of fermented food. So let's begin with the first video. But before that, make sure to subscribe to this channel and comment I subscribe. Okay, so introduction to the microbial world. Here we are, and if we start, we have to know the concept of microorganism. It's a living being smaller than 1 millimeter. And they are named and classified. We have the bacteria, the fungi, the protozoa, the algae, and the viruses. And they have a specific microbial taxonomy. We have the domain bacteria, the domain archaea, the domain eukarya, viruses, and subviral particles. Indeed, a very important thing we have to differentiate is between prions and viruses. Prions are misfolded glycoproteins. That means that instead of having a lot of alpha helix, they have a lot of beta sheet in them, and this give to the prions very interesting characteristic that we will discuss in next videos. And then the virus and the virion. The virion is the extracellular version of the virus, and once it gets inside a cell, for example a bacteria, well, we have the virus. Structure and function of the bacterial cell. Well, in terms of cell morphology and size, we can differentiate between cocci and bacilli. Cocci are the round forms of the bacteria, and bacilli, they are like a rod shape. And streptococcus means a lot of cocci, one bound to the other, forming a chain. Tetrada means 4 cocci in a square, in a 2D plan. Sarcina is like a cube of 8 cocci, and Staphylococcus is like a random aggregation of cocci, for example like grape style. Bacilli, we have loose bacilli, that those can be alone by themselves, diplobacilli, to bond together, streptobacillus, strepto as in here means a chain of, so a lot of bacillus bound together, and coccobacillus, that is like a bacillus, but that is more similar to a cocci. And a very important characteristic of these bacteria is that they are very small, and as being small, they are very, very adaptable, and they can multiply faster. This is the general structure of a prokaryotic cell. We have the external structures, the cell wall, and the internal structures. So the cell wall differentiates the external from the internal. External, we have the glycocalyx, which can be either a capsule or a mucus layer. Then we have the flagella, that can be monometric, that's a single flagella. Amphitricos, that's 2 flagellas, one in each pole of the bacilli, for example, so one here and one here. The locometric, that means there are more than one flagella on the same pole. For example, from here a lot of flagella from the same pole. And peritricos, that means that all around the bacilli we have flagella, but that can be also in cocci too. We're talking about bacteria in here. So peritricus is flagella all around the structure all around the cell wall. Then we have fimbriae and pili. Fimbriae is used for junction between bacteria and pili for the transmission between them. Cell wall, well, the Peptidoglycan. This is a very important component because this will differentiate between gram-positive or gram-negative bacteria, and it serves to give strength and shape to the cell. Gram-positive bacteria have a lot of peptidoglycan, almost from 50 to 90%, and the gram-negative, they have only a few of peptidoglycan. Internal structures, well, we have the membrane, of course. This is not the membrane, this is the membrane. We have inside of the cell wall the membrane, which is a selective permeability and a metabolic center, because from the membrane the bacteria obtain energy, like if they were mitochondrions. And then the cytoplasm, and in the cytoplasm we can find the nucleoid, which is the bacterial chromosome, and the plasmids, and then the ribosome 70s, and the inclusions that can be either organic or inorganic. Then we have the endospores in some of them, and this is very important because these endospores are used as a protection the bacteria uses when the circumstances are not favorable. And the endospores can be deforming and non-deforming, either one or the other, and terminal, subterminal or central, depending on the position inside the bacteria. Then we have the microbial nutrition. Well, the chemical requirements they need, as we do, macros and micros, and they also have the growth factors. Macros of the bacteria: carbon, nitrogen, and then calcium, magnesium, potassium, sodium, and iron. To remember this very easily, just remember that these are all electrolytes: the sodium, the potassium, and even the calcium and the magnesium can function as electrolytes. And then we got the iron. If you want to know more about electrolytes and iron, check my videos on the nutrient description series where I talk about minerals. And then we have a bunch of other minerals as micros. And then the growth factors are very important, such as vitamins, amino acids, and nitrogen bases. So basically vitamins help in a lot of metabolic functions, and the amino acids, well, they have a very important growth factor because they are anabolic. And the nitrogen bases, they're used for synthesize the nucleic acids from which we can get the code to actually create those proteins and the growth. The fermentation and respiration. They can obtain ATP using both of them, either fermentation at a substrate level, and it can be lactic, alcoholic, propionic, butanoic acid mixed, or butyric. And then we have the other part, which is the respiration using the transport of the electrons. This can be either organic or inorganic, aerobic or anaerobic. They use the mechanism of the oxidative phosphorylation using the electron transport chain. As I mentioned previously, they use the membrane as a metabolic center, and they basically use the electron transport chain in there with all the complex we viewed in the video of the electron transport chain in my channel. And then the environmental requirements, yes, because these were the fundamentals they need inside to live. This is the energy, so these are like the food, the energy, and this is the environment. We have oxygen, Some bacteria are strict aerobic. That means that they need a 20% of oxygen in the air. Then there are the microaerophilic, which only need from 2 to 10%. And then the facultative anaerobes or facultative aerobes that can live either with or without the oxygen. Then the aerotolerant anaerobes, that they are mainly anaerobes but they can tolerate some oxygen. And then the strict anaerobes, that the oxygen is actually harmful for them. Then the temperature, we have the secrophiles that like being cold, like close to 0 degrees Celsius. The secrotolerants, that they grow mainly at 15 degrees, but they can also grow at 0. And the mesophiles between 20 and 45 degrees Celsius, the thermophiles 60 degrees, and the hyperthermophiles 100 degrees Celsius. In terms of pH, we have the acidophils from 1 to almost 6, then the neutrophils from 6 to 8, remember the neutral is 7, and then the alkalophils from 8 to 14. In terms of sodium concentration, we have the non-halophilic that hate sodium, the halotolerant between 5 and 10% of sodium, the halophilic between 10 and 15, and the extreme halophilic between 15 and 30. Okay, fine. This was the introduction video of this series. I hope you liked. Give me a like down below. Share this video to your classmates. I know they will like this stuff. I will leave this diagram down in the description so you can have it. It's on my website. Download it. It's free. And that's it. See you on the next one. Bye.